For the gelatin test, you will need an inoculating wire, Bacto incinerator, which is right here. You will need your unknown bacteria in your slant stock culture form, which is this, and then you will need your gelatin agar. At room temperature, it is liquid, just so everybody knows. Once you have all your materials, you are ready to begin. First, we are going to sterilize our inoculating wire. To do that, stick it in the Bacto incinerator for about 5 to 10 seconds. Once you've done that, allow the wire to cool off. Once your wire has cooled off, take your test tube that contains your slant stock culture from your unknown bacteria, open the lid, flame the test tube for 5 seconds, and then stick your inoculating loop down in to collect a small amount of the culture, just by scraping it like so. Flame the test tube for another 5 seconds. Then put the cap back on. Put it back on the test tube rack. And then what you're going to do is, now that you've obtained a small bit of culture on your inoculating loop, you're going to grab your gelatin agar, take the cap off, and what you're going to do is you're going to stick your inoculating wire down the center of the gelatin, pull it out straight, put the cap back on, put the test tube back on the test tube rack, sterilize your inoculating loop for about 5 to 10 seconds, and then put it down on your workbench. And what you're going to do is take your gelatin, which, like I said earlier, is in liquid form, and put it on ice for 5 minutes. For time's sake, after 5 minutes, you can now analyze and examine your specimen. After analyzing your specimen, make sure that you disinfect yourself, your workspace, and also to dispose of the gelatin culture in the biohazardous waste container. To disinfect your workspace, first you're going to move all your materials out of the way. Use germicide to spray your workspace down. Then you're going to use paper towels to wipe and dry off your workspace. Once you've done that, you can throw the paper towels away in the biohazardous waste container. Once you've done that, you may remove your gloves and dispose of those as well in the biohazardous waste container. Then, go wash your hands. After you've washed your hands, grab a new pair of gloves because we will be moving on to the next experiment, which is the catalyst test.